Before we start the lecture, I just wanted to make a couple of quick announcements. First, I just want to reiterate that it is CSU's policy that we wear masks indoors and that we wear them to cover both our mouth and our nose. I really appreciate that so many of you are doing that. We want to keep everyone safe because we want to keep it in person and not have to go back online. I know it's tough. I stand up here for 2 hours talking into this thing. It's hot, it sucks, but it's what we have to do. So if we see you without a mask or without your mask properly covering your face, we'll ask you to please correct that. And if you won't do that, then we'll ask you to leave. Remember that we do stream this and we do post the recording as well. So if you can't or won't wear a mask, or if you're feeling sick at all, please just stay home and watch it from there. The second announcement I wanted to make is some questions came up about whether we would have lecture tomorrow. Yes, we will. They're not shutting the campus down until 2 o'clock, so we will be out of here in time to still have lecture. You will, however, have off on Monday, so you'll still get a nice 3-day weekend to recover from the game. Okay. Some people apparently don't even know that we're closing campus at 2 tomorrow. Yeah, I'm seeing a lot of what? Okay. All right, guys, calm down. So there's a game tomorrow, and they are going to be closing campus at 2 p.m. I believe they're closing all traffic patterns at 4 p.m., so don't be near campus around 4. You won't be able to get out.
Are they going to do that for all home games?
If it's on a weekday, they do that, yes.
Thank you. So we have class after 2 p.m., does that mean it's probably canceled?
Anything you have after 2 is probably canceled or it's online. Okay. Check with your class. All right. Guys, yesterday we finished talking about moving things from the endoplasmic reticulum to the Golgi apparatus and then to the plasma membrane. When we move things out of the cell, so we take it to the plasma membrane and we merge those vesicles, that's exocytosis. So we showed you this yesterday, but I didn't actually tell you that was exocytosis. So we'll review that real quickly just to go over it. Then we're going to talk about lysosomes. That was the other place that we could be sending vesicles from the endoplasmic reticulum to the Golgi apparatus to the lysosome. They're specialized in that they are a garbage disposal. So they can destroy all kinds of things in our cell or from outside our cell, like pathogens. They have a pH down of about 4.5 or 4.25, I should say, and then lysosomal proteins that can help them to break all the different components down. Then we'll do a brief discussion of endocytosis, which is bringing things into the cell. So you grab it from the outside, you put it into a vesicle, and you move it into the cell. We're not going to get into the mechanics of it, but we're going to point out how this is probably how we ended up with mitochondria, which is really just a modified bacteria. This is really important in our production of energy and ATP. We're going to start talking about where we can use ATP today when we talk about motors, but we use it a lot in our body. So this production of ATP is really important to us. And then finally, we'll talk about how mitochondria can be tracked through the maternal lineage. Once we've finished with the mitochondria, that's going to be all the organelles that we're going to discuss. But we can still talk about other cellular components, including cytoskeletal proteins. These are polymerized, which means that they're made from smaller bits. You take a monomer, a single unit of something, and you put a whole bunch of them together, and now you have a polymer. So these polymers are going to be important for both transport and structure to the cell. The different types are going to be microtubules. They're the largest at about 25 nanometers. Those are going to be important for the transport. These are actually the ones we're going to use primarily for moving things from the ER to the Golgi to the plasma membrane. Then there's intermediate filaments, so named because they're intermediate in size. They're about 14 nanometers. These are mostly structure. Okay, they give form and stability to your cells. We're not really going to talk about that. There's not much more to talk about. They give form and stability. We're done. Then we'll get into filamentous actin. This is going to be transport again. These are just much, much smaller than anything else. And so when we talk about those two transport cytoskeletons, again, we're going to talk about how they're polymerized from their monomer forms. Microtubules are specifically tubulin monomers polymerized into that microtubule. And then these are going to be doing long distance transport. And it's going to have, if you have these highways, you have to have something to ride on them and carry your passengers. We're going to have these molecular motors They're ATPases. They're going to break down ATP in order to move the vesicle across these microtubules. We have two different types of these molecular ATPases for microtubules. One is kinesin. It's going to move in a specific direction. It's one way only. And the second is dynein, moving in the opposite direction. Probably won't get to dynein, but we'll try. All right, so for exocytosis, we had our cell, and we had our nucleus. And to save some time, I'm just going to do this real rough endoplasmic reticulum. So now we have a pretty flower. You're going to have your Golgi apparatus. And then we said that when you're making these proteins, they're going to move all the way through the Golgi from cis to trans. And they're going to end up in this trans portion, which will then create a vesicle to move them. So if this one is a secreted protein, then you're going to move this vesicle to the plasma membrane. So you can merge this now. And the secreted protein will end up out here. The process of merging vesicles with the plasma membrane wall is what we call exocytosis. We're leaving the cell. Now, I showed a secreted protein in the example, but that could very easily have also had a transmembrane protein in the membrane of the vesicle. That's still going to be considered exocytosis, even though at the end that protein is going to be in the plasma membrane and not actually outside the cell. It's the process of merging that vesicle with the membrane that we consider exocytosis. So you can say you exocytosed that transmembrane protein. All right. The other thing we can have coming out of here was a vesicle carrying a lysosomal protein to the lysosome. And so it'll merge here and put that lysosomal protein into the lysosome. Now, a lysosome again is a garbage disposal. It can get rid of anything unneeded, recycle all the stuff that's within your cell, but it can also recycle stuff that you bring in from outside your cell. This is where you can send things like pathogens, microbes that you pick up to destroy. One of the reasons is we have all these lysosomal proteins. And I won't ask you to know these names, I just want to give you the examples. We'll have things like a protease. Okay, just so you know, anytime you see ACE, that means it's an enzyme. It's a protein that breaks things. Protease means it breaks proteins. You can have lipase, meaning it breaks lipids. You can have saccharidases, which means it breaks sugars. You can have nucleases, breaking DNA and RNA. Okay, so all of these proteins can function to break down anything you send to it, including that pathogen. You can break down the membrane with the lipase. You can break down all of your proteins and the sugars that were attached to them, and you can get rid of all the DNA and RNA that came in. Right? The other thing to note about this is that the pH is about 4 to 5. Okay. Just a quick review for those of you who haven't had this in a while. pH is a measure of your hydrogen ion concentration. Actually, I should say it's the inverse. It's measured on a scale of 0 to 14, with 7 being neutral, and anything below that considered acidic. Anything above is basic. So when we tell you that the pH in the lysosome is 4 to 5, that means it's very acidic. It's not the most acidic thing in our body. Our stomach has that honor, 2 to 3. But it's still way more acidic than most of our cell. Because the typical cytoplasm and extracellular pH is going to be about 7.2. We talked about how with proteins we have to fold them into its 3D shape for it to be functional. And we said we use bonds between either the backbones or those R groups in order to make those folds. Those are meant to work, for the most part, unless they're lysosomal proteins. They're meant to work in a 7.2 pH. If you put a protein that's meant to work in 7.2 in an environment where the pH is down to 4 or 5, it's going to interrupt those bonds. That protein will no longer be able to maintain its 3D shape, and it will unfold. We call that denaturation. So this is often going to cause a denaturation or unfolding of those proteins. Once you've unfolded that protein, now the protease can come in and clip the peptide bonds between the amino acids. And now you're back down to just amino acids that you can reuse in your own body to make more proteins. All right. Any questions on to lysosome? Yes?
So is the lysosomal proteins, are those the ones that are that come from the Golgi, are they being broken down in the lysosome, or are they like the ones being breaking down?
That's a great question. So we're talking about how we can break down all these proteins by denaturing them and then chopping them up. But we're also telling you that we're making these proteins in the ER, sending them to the Golgi, and then sending them to the lysosome. Why? So we can just chop them up? These proteins are meant to work at a pH of 4 to 5. I keep saying 4.5. Okay, that's their ideal environment. They take their proper shape and actually function best, are most active when they are here in the lysosome. So they're fine. They will not get broken down. Okay. All right, so let's talk about endocytosis. So in endocytosis, I'm going to use a different type of cell for an example. We're not actually going to talk about this cell until we get to the third unit. You don't have to know anything specific about it for this unit. This is just our example. This is a macrophage, and it's an immune cell. And its name tells you it's a big eater. So anytime you see phage, that means to eat. So I like to think of macrophages as Pac-Man. They just go around and they try to gobble up all the pathogens they can find and eat them. So endocytosis is really eating. Okay? This is you're picking something up from outside of the cell and bringing it in. So this macrophage doesn't actually look like Pac-Man, unfortunately. But it will have these receptors on it. And these receptors can find these pathogens and stick to them. So this is going to be a bacterium. Endocytosis is really the opposite of exocytosis. Mechanistically, it's not. It's totally different. But just in terms of One was a vesicle that merged into a membrane to release its contents. Now we're going to take contents and stuff them into a vesicle and bring them in. We just endocytosed both the receptor and the bacterium that it was holding on to. The goal here is to send it to the lysosome so that it can be destroyed. If it thinks this is a possible pathogen, it wants to break it down so it's not a threat. But then it's going to take the pieces of it and it's going to keep those. It's going to show those around like a wanted poster and say, hey, did you guys see this? Let me know. We need this guy. He's bad. This doesn't always work the way we expect. Because any successful bacteria or virus is one that's going to get out of this. It's not going to get destroyed or wouldn't have been able to do any damage. So these guys are pretty masterful. They can get out of that vesicle and get into the cytoplasm. Sometimes they can even get out of the lysosome. Occasionally, they'll even stay in the lysosome. Tuberculosis, TB, they're actually quite comfortable in the lysosome. So they'll get taken in, and they will live in a lysosome of your cell for decades. They'll wait until your immune system is weak, whether you get sick or whether you get older and it just weakens with time, and then they'll pop back out and take over. I'm telling you this and using this example because I want to kind of give you a background of how we ended up with mitochondria in the first place. Okay. So I'm sure this probably happened plenty of times where these things escape. We know they do it all the time because we sometimes get sick. But sometimes it's not a pathogen. Sometimes it's actually something beneficial, something we can work symbiotically with. And there's plenty of bacteria that we're symbiotic with. So billions and billions of years ago, let me see, we had these plant cells. Don't worry, I won't ever ask you anything about plant cells, it's just an example. But this plant cell picked up a specific bacteria called the cyanobacteria. This eventually became a chlorophyll. Cyanobacteria are able to use sunlight to make energy. And as an unfortunate byproduct from their perspective, it makes oxygen. Since they don't care about the oxygen, they push it out, which is great because now we can use it. But back then we didn't exist. And back then there wasn't much oxygen in the air. Now our atmosphere is something like 80% nitrogen and 20% oxygen. But when this happened, it was almost non-existent. Over millions and millions of years of more of these plant cells picking up chlorophyll and using them, we pushed enough O2 out into the system. We, like I'm a plant. They pushed enough O2 out into the system to change our atmosphere. You had a question?
So on the cell, is that a certain kind of receptor?
Probably, but we won't name it. That's an immunology class. Okay, so now we've changed this atmosphere, which sounds great to us now. But I mean, think about it. Like if now we suddenly changed the atmosphere and started adding sulfur, that wouldn't be good for us. Back then, everything that lived was used to not having oxygen. They weren't adapted for it. So it was actually a bit of a problem for most of them. It was adapt or die. And there were some animal cells that managed to adapt. So we had an animal cell. Picked up a mitochondria, And the reason these guys were so special is because they could use that O2. And they turned it into ATP. Excuse me. ATP is our currency of the cells. You'll hear that said a lot. It's energy. We use it for so many things. And if you haven't seen ATP before, it's just been a hot minute. Let's go over it real quick. This is adenosine triphosphate. Okay, and I probably should have looked this up between classes, but I still don't know the chemical structure of adenosine. I would have to Google it to print it. I would certainly never ask you to ever draw it. Okay, so I'm not going to try now. I'm just going to say that this is an adenosine attached to 3 phosphates. Okay, there's your molecule. Chemistry teachers worldwide are croaking. This is really important because the bonds between these phosphates are what we consider to be high energy. So if you break these bonds, you release a lot of energy that you can use to do things in your cells. So we have these things called ATPases. Remember that the ACE tells you it's going to break something. This is breaking the ATP. Specifically, this ATPase is going to break this last bond. It's going to cause this ATP to become an ADP, as in adenosine diphosphate, plus that broken off phosphate. So it's ADP. Okay. So we're going to talk about ATPases a lot, including today. That's why I wanted to just go through that with you real quick. Okay, the last thing I want to talk about is inheritance. I'm going to erase this. So mitochondria is interesting. Actually, nope, let's back it up. I can't talk about that until I tell you more about why or how we figured out that mitochondria was a bacteria in the first place. Because we didn't always know that. I think back in the 50s or 60s, they didn't know that mitochondria had bacterial origin. It seems so obvious now, I don't know how they didn't, but in the 60s, the scientist was looking at the cell, and specifically the mitochondria. She was interested in that. And one of the things that she noticed was that the mitochondria was very, very unusual for a eukaryotic organelle. Because that mitochondria has a double membrane. We don't see double membranes in eukaryotic organelles. You do occasionally see them in bacteria. So that was kind of her first indication that maybe this is bacterial origin. So she studied it for a while. She looked at these for years. She did all kinds of tests on them. And she found that not only do they have the double membrane, they also have their own DNA. Okay, all of our DNA in our cells should be in our nucleus. None of our organelles should just be holding on to DNA. But here's mitochondria in the middle of our cell, and it has its own DNA. It doesn't match ours. It's different. And to top it off, that DNA is circular. So we know that's not how we store it. We store our DNA wrapped on histones as chromatids. The last thing that she was able to find was that this mitochondria has its own ribosomes. The thing is, with ribosomes, there are actually differences in size between eukaryotic and prokaryotic ribosomes. We won't ever get into that detail, but it is something that you can look at a ribosome and tell if it came from a eukaryotic organism or a prokaryotic. Mitochondria have prokaryotic ribosomes. Okay. So that was the evidence that this is actually from a bacteria. And we say that it's modified because it's actually been here for so long that even though it has its own DNA, and again, don't memorize these numbers, but its DNA contains about 40 genes. Now, bacteria often have less genes than we do, but they're still typically in the thousands. 40 genes is not enough genes to survive on, especially since these were specifically like RNA-related genes. And the reason it doesn't need any more genes is because it has lived in eukaryotic cells long enough that it's able to dump all of the duplicate genes it doesn't need. If our cells make proteins it needs, it just uses those. These 40 genes are specifically for proteins that the eukaryotic cells don't make, and it still needs. Okay. Again, that part's just detail. Understand what differentiates the mitochondria from other eukaryotic organelles and why it's believed to be of bacterial origin. Okay, let's talk about inheritance. So when we create an embryo, we're going to take an ovum, and this is not repro. Repro's at the end of the class. I'm not going to get into it. We're just going to give this quick little, here's an egg. It's fairly large. It has everything this embryo would need. It's got the cytoplasm, it's got the mitochondria, it's got all the organelles. The only thing this is missing is 23 chromosomes. This only has 23 from mom. So it needs another 23. And that's what males provide. That's what sperm is. Just DNA. That's all sperm provides to this. It's just the DNA. Everything else is in the egg already. So when you have a sperm, it's tiny in comparison because it's just really a DNA transport. Now, in order to get the DNA to the egg, you have to have this tail and some mitochondria to produce ATP to power that tail. The mitochondria that's in this egg came from mom, because that's where the egg was from. The mitochondria that's here on the sperm came from dad. However, when a sperm joins with an egg, I should draw it like that. It's usually just the head that breaks through and then releases the DNA that was in the head into that egg. Nothing else gets in. So the mitochondria that's attached here, this paternal mitochondria, never gets in there. The only mitochondria that this egg has is from mom. So mitochondria is always a maternal lineage. Okay. That means if you have siblings, you have the exact same mitochondria because you both got it from your mom. Your mitochondria will not match your dad's. Your dad's will match his sisters and his mothers and his brothers, but not his father's. So you can actually track this back. And it's pretty fascinating what they can do tracking this back. They can look at mutations and where people have moved to be able to figure out how these mutations came up in the first place. But again, beyond this course, just interesting. Do know the maternal lineage. That's something we could definitely test you on. We can give you a whole bunch of people and tell you how they're related and then say who has the same mitochondria. Question in the back.
I know if they have like ATP injections now, and is that like, why is that a thing? Like, why do people sometimes have like not enough ATP? And is that inherited? Is that like, will that forever be passed down? Is that a mitochondrial?
I have never heard of ATP injections. That's the first on me. If someone didn't have enough ATP, it would suggest that it's mitochondria, since that's where your ATP is coming from. But I haven't heard of that. I'm going to have to go look that up. Sorry. Nothing more on that. Okay, if there's no more questions, we'll move on now to cytoskeleton. So, again, cytoskeleton is a major component of your cell, but it's not an organelle, it's a protein that's been polymerized from some sort of monomer. We're only going to focus on those cytoskeletal proteins that are integral in transport. So that's going to be your microtubules and your filamentous actin. We'll start with the microtubules. We'll get to filamentous actin tomorrow. But the microtubules are going to be coming from a monomer of tubulin. So you have your microtubule. It's going to be polymerized from tubulin monomers. All right. Tubulin exists in a dimer form. Di meaning two. So that means that there's two subunits that create this monomer. It's not functional unless it has this dimer form. And it kind of looks like a barbell. So you have an alpha subunit and a beta subunit. That's your single tubulin. You put a whole bunch of these together and you can make microtubule. But you have to make it in a prescribed manner. These tubulins can be self-assembled, both end to end and side to side. What I mean is, if you're looking at this tubulin here and you have an alpha and the beta, you can add one end to end, always oriented where the alpha and the beta will be connected. You can also add it side to side. That was terrible. When you do it side to side, it's going to end up slightly offset because, again, you want to make sure that you have your alpha and beta subunits in connection. So your next row would be alpha and beta. And then, of course, you can just continue bringing these out. And fill that all in. You're not just going to keep growing this forever. You're trying to make a tube that things can walk on and move vesicles. Go ahead.
So you couldn't ever like flip it upside down so it would be straight line with like alpha, beta, alpha, beta?
I mean, you could turn this whole thing.
Okay, but it couldn't ever like line up straight up and down?
Oh, so like here.
Yes.
You wanted to know if this could ever line up. Not exactly. I mean, I'm exaggerating how far it's offset, to be honest. But you always want the alpha to be next to the betas. So you always have to have them off a little bit from the one previous. So when you make them, they'll actually end up slightly circular. So what we're doing here when we do this side to side, if this is your dimer and this is your alpha and your beta is my palm versus fingers, and you take your next one, when you put them together, they're going to bind in such a way that it causes a slight curvature. I can't draw it, that's why I'm showing you my hands. When you start adding more and more on, they'll continue to curve, okay? And eventually they will come back around and meet. You can make a full radius with 13 of these tubulins. Good. Okay, so your microtubule is essentially a tube made of 13 tubulin, I can't even make it, there we go, circle, that are going around, and then you just keep tacking 13 more on, essentially.
So what are they transporting?
All kinds of things in the cell. Who asked that? Sorry. I don't know who I'm looking at. Okay, hi. They're definitely going to be transporting all the vesicles that we've talked about, carrying proteins. In neuro, we will talk about instances where they can transport things not in vesicles as well. Local transport.
And can you define dimer and polymerized real quick?
Yes, so the question was, can we talk about what a dimer is? And a dimer means that it's got 2 units to it. So this is going to have an alpha and beta subunit. That's your dimer. Polymerization just means that you're taking monomers of something like tubulin and adding them together to make a polymer, mini. So these tubulin are the monomer of the polymerized microtubule.
Thank you.
Yes?
Where do these microtubules exist in the cell?
That is a great question. Where are these in the cell? Let's look at the cell real quick. Okay. Here's a microtubule. Here's a microtubule. There's a microtubule here, here, here. In other words, everywhere. Okay, this is a major component of cells. If you look at a cell, and I'm going to show you a really pretty picture tomorrow, you're going to see that there's just microtubules everywhere. This is transport for the entire cell. Yes?
So when you say the radius is 13 tubulin, do you mean like from the nucleus like out, or do you mean like in?
I'll show you a pretty picture. It's not as pretty as the one I'm going to show you tomorrow, but it may be helpful in terms of visualizing the microtubule radius versus length. Yes? Somebody else had a question?
Do they exist in the extracellular space as well?
So no. And this is a really good point. The question was, is microtubules in the extracellular space? Microtubules are cytoplasmic proteins. They stay in the cell. So when you make these tubulins, would you make these on bound ribosomes or free ribosomes?
Free.
Free, thank you. These are ones that are going to be made on free ribosomes because they work in the cell. Okay, so this is a picture that hopefully helps. Here's the 13 that are lining up to make that full radius. That's like if you take this microtubule here and you just turn it up so that the microtubule is running into and out of the board. There is a tunnel in between. We don't use it. That's not how we're moving things. We move things on top of the microtubule. So it's actually running on this. And here you can see, let's see, the light green is the alpha subunit and the beta subunit. So you can see how they're slightly off center. So if you followed it around, it does kind of spiral. Okay. All right. So the other thing that's really important about microtubule, as well as filamentous actin, is that it has an endedness, meaning that just like we orient this always alpha to beta, the sides that have the alphas on them are what we call the minus end. We call that the minus end specifically because we can subtract from here. You can actually start taking these tubulins away. So if you have your microtubule and you're trying to shorten it, you take this one away, then this one, then this one, then this one, down to 13, come back and start again. This is the end you're going to remove them from to make it shorter. You want to make it longer, you add them. And you're going to add on the plus end. So you can imagine if you just had this microtubule in your cell somewhere and you wanted to move it, you don't just have to take the whole thing and move it. You literally treadmill it, we call it. You'll pull off from the one side and add to the other, and when you're done, it's over here now. This orientation is also really important in terms of those motors because, like I said, the kinesin and the dynein both only go one way. Kinesin towards the plus end, dynein towards the minus end. Question?
Is the plus end noticed by the beta?
Is the plus end? Yes. That's a great question. So the plus end is always going to be the side that the beta is facing. Okay, I know I didn't fill this in very well, but you're always going to be adding on as an alpha onto an existing beta. You're always going to subtract off from the side where alpha is. And let me show you that orientation in the cell. So here was a microtubule. This is the minus end. The minus end is always facing towards the nucleus. This side is the plus. The plus side is always facing towards the plasma membrane. Okay. All right, let's talk about the motor that works on it. So we've now made all these microtubules. Now we need a car. And we'll start with kinesin. Kinesin is the best well-studied of these. It's also kind of the neatest. There's a video under Tools for Success of kinesin walking like an actual, it's adorable. Kinesin is plus end directed, meaning that it will always be walking from the nucleus towards the plasma membrane. Walks away from the nucleus. Kinesin is going to have these little feet that are connected to these alpha helical coils. And then it has a head. This head in blue we call an adapter protein. And that's what's going to hold the vesicles, or it'll hold the cargo, we'll say. We're going to show a vesicle with proteins in it. Okay. If I was kinesin, my arms would be the adapter proteins, and I could hold cargo and carry it. Those alpha helical coiled structures would be my legs. And then the feet are the actual ATPases, and they'd be my feet. So these guys are the portion that are going to break down ATP and cause movement. So let me draw a microtubule here for him to walk on. And I'm just going to kind of draw in some of these dimers. Okay. He's going to have ADP in these binding pockets. I don't know why I called him a he, but we'll name him Kenny. Kenny has ATP diphosphate. I always have trouble like pronouncing D and P, so sometimes I'll say di and try to make it clear. This is going to be an adenosine diphosphate in its feet, in its little binding pocket. It will always have ADP bound when it's at rest. So this guy actually does get breaks on occasion. He'll just float around the cytoplasm looking for more vesicles or cargo to carry. And when he finally does pick something up, he's going to look for a microtubule to walk on. When he finds the microtubule, one of these feet can come in and bind specifically to the beta subunit of this dimer. So this foot can come down here and bind. When the foot binds, Just like when you put your foot down, it changes shape slightly. This will kick out the ADP. Now you have an empty binding pocket, and ATP can come in. Now we're making ATP all the time in the mitochondria, and we just put it out in the cell, so it's out there. As soon as that binding pocket's available, the ATP will come in. So now we're going to put ATP in there. Now, it's a little difficult to describe just because there's certain things going on at the same time, but I will try to walk through this, pun intended. Your ATP, when it binds, is going to cause something called a conformational change. We're going to use that term a lot. Conformational change means that when you're binding something, the binding itself is going to change the shape of the protein. In other words, like if you have hydrogen bonds now, it will pull the protein a little tighter, and it changes the shape. This is attached to those coils. If you change the shape of this foot a little bit, it's going to cause that coil to turn. Turning that coil is going to cause this back foot that was not bound to whip forward. So this guy now is going to whip over here so that it's in line with the next beta subunit. So here's our other foot. This still has ADP into it, which makes it sticky. I should say that we like to say that ADP is sticky. So it makes it good for sticking to the microtubule. This ATP is going to get cleaved pretty quickly. That's what this is. It's an ATPase. That's its job. So it's going to cleave this ATP into ADP. That's going to cause another change in shape. It's going to release this foot. So now this foot's up here with ADP bound to it. This foot, because it's now flattened out a bit, drops that ADP out. Another molecule of ATP that was floating around can now get in there. Again, that's going to cause that conformational change that will cause the coils to shift. Where are we? Green foot now will flip forward. And so it can just continue to do this down every single beta. One foot there. I should do this as like orange and green. You'll move one foot here. The green foot will come here, then orange. And so it just walks all the way down. And I'll show you a little example. Oh, we got like 2 minutes. I can do this. So again, I'm kinesing. I'm holding a vesicle. Here's my little vesicle. I've got my feet. I've got ADP on them. That ATP can stick to the beta subunit. When I stand, the ADP leaves. ATP comes in. Now I can't do coiled, I'm not that flexible. So we'll just pretend. But the ATP will cause it to change enough that this flips forward and can stick to the next beta. The ATP here cleaves to ADP, pops up. Okay. This is going to now leave the ADP. ATP comes in, coils, causes this to switch forward, and I'm just going to walk my little vesicle down the whole microtubule. Okay? And we'll talk about dining tomorrow. Thank you.